Hi, my name is Thomas Marin. I'm a first year PhD student on the ICB Centre of Doctoral Training Programme funded by the EPSRC. I work across the groups of Professor Vila, Professor Tate, and Dr. D'Antonio, and in this video I'll be briefly speaking to you about the research I aim to complete in my PhD on developing novel methods to interrogate the quadruplex DNA interactome in live cells. GA-rich sequences of DNA are capable of self-association and forming non-canonical secondary structures called G4s. Hydrogen bonding of the hosting phase of each G nucleobase enables these four-stranded structures to readily form coplanar G tetrads under physiological conditions. Two or more G tetrads pi-pi stack in forming a G4 structure, with additional stabilization derived from coordination of a monovalent cation, typically potassium or sodium. Dependent on the conditions with which they form, strand directions, loop lengths, and composition, G4s can adopt a range of intramolecular or intermolecular topologies. These G4 structure motifs have been associated with numerous biological processes such as transcription, replication, and genome instability, among others, while they are also implicated with various diseases, primarily cancer, but also neurodegenerative and age-related disorders. Given the importance of these structures in such a range of biological processes, understanding how G4 structures interact with their intracellular environment is critical in elucidating G4 function and the principal regulatory mechanisms of these interaction networks. Therefore, my PhD will focus on developing methods to identify protein G4 interactions in the native environment. Herein, we propose using photoactivatable ligands to bind to G4s before light activation generates a reactive species capable of spatially selective labeling of nearby interacting proteins. Through subsequent experimental workflows, we are then able to isolate and identify these labeled proteins, thereby enabling us to understand these molecular interactions, which will help us to unravel the mechanisms by which G4 structures mediate a range of biological processes and diseases they are implicated with. Thank you.